Thank you for the opportunity to give you an update on the work we've been doing to try and progress a gene therapeutic approach for DADA2. I'm doing this talk on behalf of a number of people. Paul Brogan, Ying Hong are sitting in the audience, and there's a number of other people thinking about this project. We all are based at UCL Great Ormond Street Hospital Institute of Child Health in London, UK. So I think we have heard a lot over the previous conferences and today about the genetic cause of DADA2 and some talks about genotype-phenotype correlation. I think we've come a long way in terms of understanding the underlying pathomechanisms. Choose your favorite. I'm glad I left the actual squares there empty. I think it's obvious that there's probably more than one mechanism and more than one function for the ADA2 protein. And we certainly now know that there's specific therapies that work very well for some of the manifestations of the disease. So we know that TNF inhibition works well for the vasculitic and autoinflammatory components of the disease. But I welcome the opportunity to look at the long-term, really, prognosis of some of these patients and what happens over time. Is it the case that some lose efficacy in terms even for vasculitis and autoinflammation in terms of response to TNF inhibition? And we certainly know that there's still patients, particularly those with bone marrow manifestations, other hematological manifestations and immunodeficiency that don't respond very well. And these are usually candidates for stem cell transplantation. Well, that's obviously if you have the right donor and with all the known caveats about mortality and GVHD rates. So given that we know the genetic cause for this disease, could a genetic therapy be a possible cure? Can we dare to use the word cure for this disease? In addition, some other favorable arguments, the fact that stem cell transplantation, that's also targeting CD34 stem cells, the target cell for gene therapy as well, is successful, makes us think that this approach could be really an option for our patients. And protocols already exist and have been taken all the way to actual clinical trials in humans for ex vivo viral delivery to stem cells for another disease, ADA1 SCID. This is obviously the genetic deficiency of an isoenzyme ADA1 that acts in the same pathway as ADA2. So on the background of this, we have hypothesized, and this has led to this program of work, that using gene transfer strategies, it is possible to correct the defects associated with DADA2. I wanted to explain some very general principles of the approach we've taken. So really, very firstly, we've taken an approach that would be suitable when we make it to clinical trials for all the patients, irrespective of the actual mutation they have. So the idea here is that we've got a mutant gene, so the mutant ADA2 gene, and we're following an approach of gene replacement addition, so we're giving a whole correct functional copy of the ADA2 gene to the cells. And this would be an actual ex vivo autologous gene therapy approach. So the idea will be that you take the stem cells from the patient, you then have your therapeutic transgene, in this case for ADA2, packaged within a viral vector that then is transducing your stem cells outside the patient's body. You expand them and then you re-administer these gene-corrected cells into the patient. So an ex vivo autologous gene therapy approach. There's a number of different viral vectors one can use. We're working with lentiviral vectors, a third-generation lentiviral vector construct, and I'll tell you a bit more about the exact details of the vector we've got. The advantages in general are that these lentiviral vectors have been the primary vectors used for stem cells for a long time now. They've got prolonged stable expression, and they're usually integrating within the genome. They're highly efficient, and they've been modified to be less immunogenic and are self-inactivating. And as we said, there's a solid track record of using such vectors for actual other human diseases. ADA1 SCID is an example. And I think an additional advantage that quiescent cells can actually be transduced. So let's look at some of the results we've obtained so far and some thoughts about the future direction. We published our data in Frontiers of Immunology last year. Ying Hong, as I said, is the senior scientist who's working on this project, and the majority of the data I'm presenting today have been generated by Ying. So in the first instance, in order to really decide what would be the promoter we would use for these cells, the actual lentiviral vector, we looked at gene expression across different immune cell types. And as others have shown, we could see that ADA2 was expressed not only in monocytes, but other immune cells, so T cells, B cells were also expressing ADA2. So we've chosen to work with this promoter called EFS, which is a ubiquitous promoter, and actually could target all these different types of immune cells. In the first instance, we wanted to explore whether healthy CD34 cells, when they're transduced with this ADA2 encoding lentiviral vector, were behaving healthily. So we looked initially at their potential to proliferate, so that's this graph, the first graph, and then their ability to form colony-forming units across the different myeloid and erythroid lineages. And as you can see, the healthy cells were tolerating this process of lentiviral transduction well. We could also see induction of ADA2 activity as shown in the last graph. We then looked at obtaining some CD34 cells directly from the marrow of a patient who had ADA2, who we were treating with red cell aplasia. So initially, the untransduced cells had very poor proliferative potential, but then you can see that post-gene correction, this was improving, and the same was the case for colony-forming units across, again, myeloid and erythroid lineages. Then we looked at whether we were recovering expression of protein ADA2, and you can see in this plot that this was indeed the case for gene-corrected cells. And the same for enzyme activity, we were recovering enzyme activity post-gene correction. The next question we asked was looking at inflammatory release cytokines released by macrophages derived from these transduced cells. And we looked at TNF-alpha, we looked at interleukin-6, and we looked at interferon gamma. And again, you can see from in this graphs that we were getting downregulation of this inflammatory cytokines post-gene correction. We also obtained macrophages from a number of other patients with ADA2, and we looked at lentiviral transduction, what was happening post-gene correction in terms of the protein expression, and this was well recovered. The same for ADA2 enzyme activity. Again, for TNF-alpha, interferon gamma, and this time we also looked at p-STAT1 signaling. You could see either in the pooled data or for individual patients that actually we were getting downregulation of all these inflammatory readouts post-gene correction. We also tried to answer the question about interaction with endothelial cells. So that's the last graph you're looking at here. So we took macrophages from these transduced CD34 cells and co-cultured them with endothelial cells. I looked at CD62 as an expression of endothelial activation. So uncorrected cells, we could see were inducing endothelial activation. But when you were gene correcting, the endothelial marker CD62 was coming down. In terms of cell lines, we heard a lot about cell lines today. We also had previously shown you data about an ADA2 knockout cell line in THP1 cells. We could also see that when you were actually introducing a working copy of a wild-type healthy protein, you were recovering protein expression and also enzyme activity, and TNF levels were coming down. But when you were inserting proteins, mutant proteins, there was some expression, variable expression, no enzyme activity, and TNF-alpha levels continued to be high. We're in the process of completing some in vivo studies. Obviously, we don't have an animal model, so we're using NSG mice. These are immunodeficient mice that are susceptible to human CD34 cell transplants. And we're transplanting them with gene-corrected cells and mock-corrected cells. And we're looking at a number of readouts over time that have to do with hemopoietic organs. So we're looking at biodistribution, engraftment, some ADA2 readouts, and also looking at histopathology, but also we'll explore what's happening to non-hemopoietic organs. We are also trying to take this forward in terms of being trial ready. So we're in the process of doing this work with a company called Viva Biotech and with some generous funding we obtained recently to actually look at scaling up. So the idea here is to establish a robust GMP-compliant process in terms of manufacturing the vector. So we're ready, we've got that up and ready to go. We will also undertake some additional required safety and efficacy studies, looking at both monocytes and other immune cells. And I think we have a key question to answer about optimizing autologous, grafts. I think we need to think about which patients will be selected for the trial and ultimately which are the best patients to receive this type of treatment, protocols for cell mobilization and conditioning regimens, and are there any key ways we can help the ex vivo expansion of the stem cells. For some other hematological diseases in our institute, people have used different molecules, CD34 agonists. We need some detailed immunophenotyping to try and understand what's the nature of the CD34 cells we'll be obtaining when we're mobilizing these patients, and I know Alessandra will present some data as well from their end looking at this. So I think I often get asked, well, we got asked this question back in 2016, how long this will all take. And I think every step of the way I'm realizing that there's a lot of really things I haven't thought through and a lot of paperwork and regulatory approvals we need to obtain. So I think we sort of said initially will be 5-6 years for the preclinical stages of the work, and I think we're kind of working our way through that. I think the pre-trial scaling up, looking at all the dose effects, the GMP compliant process will probably take us another 3-4 years. And I hope we'll be ready for phase 1-2 trial by then. We often get asked also if we have considered more modern approaches of delivering the ADAR2 gene. And CRISPR-Cas9 as a platform, an editing platform that I guess in general would maintain the physiological regulation of the gene has been used for other diseases. So we have considered this and have designed some guide RNAs coupled with Cas9 that essentially will go and recognize the right bit within the DNA to induce a break. And the idea here is that we will then use a template through this process called homology-directed repair. So this is an exogenous template of healthy DNA ADAR2 that's used internally by the cell to induce repair. However, the issues that I think several others I was talking to Harry have faced before is that off-target effects remain an issue. I think viral delivery, would we be using an AAV, which we know can be immunogenic? Can we use lenti to be a bit more targeted? Are still considered. And also it seems that stem cells are very sensitive to DNA manipulation, so they're not very receptive to this CRISPR-Cas9 approach. There's issues with them in terms of stemness and fitness and engraftment. So we've got a number of steps to work our way through, but certainly we're considering this approach. I think the other question I get often asked is about base editors. Of course, there's specific base editors that have been used for T-cell malignancies. The approach would be for individual mutations, so it's not an approach that would fit all the different patients, but certainly something that we can consider. And I wanted to put this into the context of what's happening in the field of primary immunodeficiencies in terms of developing gene therapies for rare diseases. So I think we obviously have learned a lot from their experience in terms of preclinical data and clinical trial data, but we have also learned that this gene therapy approach for rare diseases needs to be sustainable and affordable. And I think the unique challenge we have is that when we're talking about small numbers of patients, it often seems that this is a too costly an approach, so getting grant funding to continue the work seems to be challenging. There's a complexity around the manufacturing process and an increased burden of preclinical testing that to me initially seemed disproportionate to what we were trying to achieve. And also there's a regular need for quality controls and regulatory assessments. So in the field of primary immunodeficiencies, there's been a number of different diseases where gene therapy products have been developed. I think Strimvelis, we mentioned that, the prototype for ADA1 SCID had achieved licensing, marketing authorization in Europe, what was an option for the patients. But then, September last year, there was discontinuation of the commercial involvement despite demand for the patients. That has obviously caused a lot of stir in the field. And it became obvious that this for-profit model approach was not fit for purpose when you wanted to develop these gene therapeutic approaches for rare diseases. This initiative is an important initiative that started from the Milan group, Professor Aiuti, and I'm sure Alessandra may mention that as well, with Claire Bruth from Great Ormond Street Hospital and Adrian Thrasher. And the idea here is to create this legal entity, they just got legal status, to facilitate access to effective therapies for treatment of ultra-rare diseases in Europe. It's a combination of stakeholders, patients, clinicians, academics, and scientists. And what's the aim here? Well, the aim here is that this trials that we're doing for rare diseases could be sponsored by this legal entity or academic organizations or hospitals, rather than really rely on marketing this and commercial interest. Have a structure that allows us to do this with minimal data and proportionate data for what we're trying to achieve, and ultimately be able to get the cells to the patients, not that the patients traveling to centers. So this would be a treatment that's available to all patients, and lobbying and education. So in summary, I hope I've shown you we're making some progress, but of course we've still got a long way to go. It certainly seems for our data that with lenti correction, we can reverse the immunophenotype of the monocytes. We're recovering protein expression and enzyme function. Let's see where we are in the next couple of years. Key questions to answer about the autologous grafts, patient selection, conditioning regimens, treatment protocols, and I think we need to ensure that we join the conversations about ensuring wide access to all patients, because this is for all the patients, and for me it should be an academic venture despite market interest and commercialization. Of course, a lot of people to thank: Paul Brogan, Ying Hong, who have been working on the project, my collaborators Claire Booth, Adrian Thrasher, and all the funders that have helped us in the DADA2 Foundation, patients and parents. Thank you.